Okay, let's continue our discussion of the biological bases of mind and behavior. First, just an announcement, or actually more like a reminder. This is the first week of discussion sections, so you should be sure to attend your discussion section this week. For people whose discussion sections were on Monday, you start next Monday. If for any reason you can't go to your discussion section this week, contact your GSI, otherwise you risk being dropped from the course. If you're waitlisted for a discussion section, you should go to the discussion section for which you're waitlisted. If there's a seat available, we'll do our best to accommodate you. Okay, in the last lecture, we began by looking at the elementary and peripheral parts of the nervous system, neurons, nerves, how the autonomic nervous system is organized, how the somatic nervous system is organized into the spinal nerves and the cranial nerves and all of that. What we want to do today, begin by turning attention to the brain itself. This happens to be Albert Einstein's brain, which was studied after he died. And at least on first appearance, the brain seems to be a single massive organ that sits on top of the spinal cord, with some other structures located underneath, which I'll talk about in a little while. But on closer examination, it's possible to differentiate among various pieces of the cerebral cortex by virtue of a couple of landmarks. First, looking at the cortex from the front or from the top, you see that the brain is divided into two hemispheres by this cerebral commissure. Just means valley, okay, right there. You can see it from the top. You can see it here from the front, which divides the brain into two hemispheres, one left and one right. These hemispheres are themselves connected by a bundle of nerve fibers that are collectively known as the corpus callosum. And this bundle of nerve fibers actually allows the two hemispheres to communicate with each other, to pass information between themselves. Then, looking from the side, we see a couple of other landmarks. Here's where the cerebral commissure is. But running right down the side is what's known as the central fissure, a very large fold in cerebral cortex. You can see it here in Einstein's brain. There it is in the left hemisphere. There it is in the right hemisphere, which separates two lobes, the frontal lobe of the brain, of cerebral cortex, from the parietal lobe. And then there's what's known as the lateral fissure, so-called because it runs back and forth here, lateral fissure. You can see it here in Einstein's brain, and again here in Einstein's brain, which separates the frontal and parietal lobes from the temporal lobes of the brain. And then finally, back here, right square in the back of the brain, is a fourth lobe known as the occipital lobe. The division between the occipital lobe and the other lobes isn't quite as nicely demarcated as these others are. There isn't anything like the central fissure or the lateral fissure, but you kind of know it when you see it. Okay, so looking from the top, the brain's divided into two hemispheres. Looking from the side, the brain's divided into four lobes: frontal lobe, parietal lobe, temporal lobe, and occipital lobe. The cerebral cortex is also sometimes known as the neocortex, neo being the Latin word for new, and the reason for that is that the cerebral cortex is the last structure in the nervous system to develop. And that's true if you consider development phylogenetically in terms of the evolution of the species. The cerebral cortex emerged relatively recently in evolutionary time. Okay? There are lots of animals that don't have much cerebral cortex. And it's also true that the cerebral cortex is neocortex ontogenetically in terms of the development of the individual species member. it turns out that the cerebral cortex develops relatively late in fetal development. Here is a diagram taken from Scientific American some years ago, comparing the cerebral cortex in various species. Here's the striped bass. You can hardly see it. The leopard frog, the grass snake, you can see a little bit of it there. Pigeon, you can see it beginning to emerge right there. The opossum, the cat, the macaque monkey, the chimpanzee, and the human, okay? One thing you notice here is that the cerebral cortex gets bigger as you proceed from one species to the next, but that's not the important thing because the body also gets bigger. What's important is that in humans and other hominid or primate species, the proportion of brain mass devoted to cerebral cortex is vastly larger than it is for non-primate, non-mammalian species. Here, relatively little brain mass is devoted to the cerebrum. When you get to the mammals, and you especially get to the primates, a lot of brain mass is devoted to cerebral cortex, so much so that in the primate species, the brain has to fold in on itself in order to get all that cerebral cortex into a relatively small space afforded by the skull. These pictures are a little misleading because the brain really isn't really a bundle of neural tissue that kind of looks like this. It's really more like a thin sheet of neural tissue. It's more like a bath towel than anything else. Some brains are more like bath towels than others. In order to get inside the skull case, it has to fold up on itself. And these folds are characteristic folds. The lateral fissure here, the central fissure, and all these other folds and protuberances. Okay, so again, viewed phylogenetically, when you get to the species that have emerged most recently in evolutionary time, you get a lot of cerebral cortex. And that's true ontogenetically too, in terms of the development of the individual fetus member. This again from Scientific American. Again, this is the neural tube of a fetus 25 days into gestation, and you really can't see any differentiating structures at all. 35 days, something's beginning to happen there. 40 days into gestation, you can see the beginnings of the cerebral cortex. 50 days, 100 days is more than 3 months into pregnancy, and that's what the cerebral cortex looks like. 5 months, 6 months, 7 months into gestation, notice that the cortex is getting so big now that it's beginning to develop those characteristic folds so it can fit inside the skull case. 8 months, and then 9 months, you see a pretty characteristic human brain with the lateral fissure very visible and the central fissure very visible. Again, the fetus is getting bigger, so it's not surprising that the brain is getting bigger. What's really interesting here is that as the fetus develops, proportionately more of brain mass is devoted to cerebral cortex compared to other kinds of structures. It's a development that's visible in proportional terms, not in terms of sheer brain mass. This is especially true when you consider a particular portion of the cerebral cortex known as the prefrontal cortex. How something can be prefrontal isn't completely clear. There's either front or it's in front of the front, right? This is the frontal lobe. Here's the cerebral commissure, lateral fissure, frontal lobe here. This portion of cerebral cortex that's way to the front of the frontal lobe. You can see that in the squirrel monkey, the cat, the rhesus monkey, the dog, there's relatively little prefrontal cortex compared to the rest of the brain. But when you get to our closest primate relative, the chimpanzee and human, a huge amount of brain mass is devoted to prefrontal cortex with implications that we'll discuss in a couple of minutes. So again, you see this principle of proportional development. Proportionally more brain mass is devoted to cerebral cortex in humans compared to other species, and that's especially the case for so-called prefrontal cortex. Proportionally more of that is devoted to in humans rather than other species. Okay, now underneath the cerebral cortex are some other structures that are important. Some of them are brainstem structures, others are so-called subcortical structures. So here's the top of the spinal cord, and you can see a structure known as the medulla, okay, which controls vegetative functions, heartbeat, digestion, respiration, things like that. We have the pons here, which helps regulate cortical arousal. Here's another structure known as the cerebellum, which is involved in coordinating sensory motor activity. And then moving up further up the brainstem, here's a structure known as the thalamus, which we'll talk about in a minute. Notice that the thalamus is up here buried right underneath the cerebral cortex. Here's where the lateral fissure would be, temporal lobe, frontal lobe, parietal lobe, occipital lobe. There are these structures, like the thalamus, that are nestled up underneath the cerebral cortex, and for that reason, they're called subcortical structures. So here we have another division of the brain, basically the hindbrain, the midbrain, and the forebrain, so called because in most animals, the quadruped animals, the hindbrain is toward the back of the animal, and the forebrain is toward the front of the animal. We walk upright, so for us, the hindbrain is low, okay, and the forebrain is high. But again, if you read in the literature on neuroscience, you'll see reference to hindbrain structures and forebrain structures and midbrain structures, and you'll want to know what that is all about. Okay, so now those are the basic features of the anatomy of the central nervous system, the brain and the brainstem. But we're doing more than anatomy here. This isn't a biology course. This is a psychology course. What we want to know really is how these various structures, how these various components of the nervous system are related to the mind and behavior. What are the psychological functions of these different kinds of structures? Well, for example, take a very dramatic loss of mental function known as coma, a loss of consciousness in which the patient does not respond at all to stimulation, shows no signs of emotion, but the individual's vegetative functions are okay. Individuals in a coma have lost consciousness, but their hearts still work, they still breathe, they still digest food and eliminate food and all of that stuff. Their eyes are closed, but they don't seem to go through the normal cycles of sleep that a person with an intact nervous system does. In patients suffering comatose patients, you see damage in one of two areas of the brain. You either see damage in the upper portion of the brainstem, which includes a set of structures known as the reticular formation because it kind of folds back on itself, okay? Or that's the red part of the brainstem, or the thalamus, here indicated in blue, which has the function of dispersing sensory information to various parts of the cortex. Damage to these areas, one or the other of these areas of the brain, will lead to a loss of consciousness. These areas of the brain seem to have, among their functions, the preservation of normal waking alertness and attentiveness. But when the damage is confined to these areas, you lose consciousness, but because there's no damage down here in these brainstem structures that mediate vegetative functions, the patient's vegetative functions will be okay. So when you look at cases like coma, persistent vegetative state, and other similar neurological syndromes, you typically see the damage in these locations. And the correlation between structure and function, the fact that you lose consciousness when you suffer damage to your reticular formation or your thalamus, is an important clue as to the function of these particular structures. Okay, they don't just sit there, they're actually doing something more or less specific, and that is to say that the function of these structures is to regulate normal waking consciousness. What this illustrates is a general principle known as localization of function. The general idea behind localization of function is that different parts of the brain are specialized to serve different psychological functions. Now, this is an old idea. It goes back to the early part of the 19th century, and a group of people known as phrenologists. It's a pseudoscience, okay? But in Europe, Gall and Spurzheim, in the United States, actually a whole family, the Fowlers and the Sizers, promoted the idea that different parts of the brain were associated with different mental functions. And they illustrated this structure-function correlation with these so-called phrenological heads, which you can sometimes find in antique shops. They're always fun. Somebody actually still makes these. So if you want to buy one on Amazon, you probably can. But according to the phrenologists, the brain was not simply a single lump of neural tissue sitting on top of the spinal cord. Instead, the brain was composed of more or less discrete mental organs, each of which was associated with some psychological function or faculty. In traditional phrenological doctrine, there were about 35 of these faculties, and if you think about it for a second, they represent a kind of common sense organization of mental life. If we sat down and took a poll and said, what are the things that people do with their minds, you'd come up with a list that's not unlike that of the phrenologists. So they thought that there was a part of the brain here, located near the bridge of the nose, that was involved in the perception of form. Here's an area of the brain they thought was involved in the perception of size. Here is a part of the brain they thought was associated with mirthfulness, kind of a sense of humor, tunefulness, musicality. Here's a part of the brain they thought was associated with cautiousness, and another one with friendship and combativeness and destructiveness, emotiveness, an interest in conjugal love, parental love, a different kind of love, self-esteem back here, benevolence, veneration. These were the kinds of functions the phrenologists had in mind, and where they located them in the brain. Now, where they get this idea? Well, what Gall and Spurzheim would do is they'd find some friend of theirs, some guy hanging around the club that was particularly high in mirthfulness. He was always laughing and giggling and telling jokes, and they'd look at his skull. And if they identified a kind of bulge someplace in his skull, they'd say, oh, that's where he gets his mirthfulness from. That must be where the mirthfulness area is. Or if they found somebody who was particularly lacking in a sense of humor, they'd again examine his skull, and maybe they'd find a depression someplace, and they'd say, oh, look, he doesn't have any brain where his mirthfulness area is. So looking at structure function that way. It was an incredibly bad way to do science. I mean, everything's wrong with it. A 6-year-old can identify the problems with this. But phrenology kind of took off as a doctrine, and it turned out that the phrenologists had the basic idea right. It turns out that the cerebral cortex isn't just a single massive organ. It is composed of a bunch of discrete centers, each associated with some psychological function, but they got the list of functions wrong, and they got the correlations with structure wrong in every detail. They didn't get one right, as far as we can tell, nor could they have except by accident, given the way they did it. So the general idea behind phrenology, which is the idea of localization of function, is right, but the details of 19th century phrenological doctrine are quite wrong. How do we do it today? Well, we have a number of different ways of studying structure-function correlations. Historically, one of the most important has been the study of patients with brain lesions. That is, patients who have suffered damage to some particular area of the brain. And what we can do is we can localize where that lesion is, what part of the brain's been damaged, and then determine what psychological functions they have lost. Usually, these lesions come about by accident. In the 19th century, the neurologists studied people with gunshot wounds to the head quite a bit. And occasionally, as you'll see in a minute, there are surgical lesions to brain tissue where surgeons have deliberately excised some portion of brain tissue for other reasons and then observed the corresponding loss of function. Another technique that's used historically is electrical stimulation of the brain, where you remember that the brain's an electrochemical system. It operates on electrical principles. So what happens in these cases is you put a microelectrode down into some portion of the brain, and you run a very weak electrical current through it, stimulating that portion of the brain, and you see what happens. Okay, so maybe you stimulate the brain and the person moves his left hand. You might think that you've hit the area that controls movement in the left hand. The most recent development in the methods for studying functional specialization are a variety of techniques that allow us to image the activity of the brain in neurologically intact, living, awake human subjects. So you can put a subject in a CT scanner and look at the structure of the brain and correlate that with function. Or right online, you can use a technique known as positron emission tomography, PET scanning, or magnetic resonance imaging, MRI scanning, to actually observe the brain working, what parts of the brain are active as subjects perform various kinds of tasks. I'm not going to spend a lot of time on this now. There's quite a bit of detail on this in Gleitman's text, especially chapter 3. And there's some detail on it in the lecture supplements for those of you who are interested. I don't want to get caught up in the various techniques. What I want to do is to give you some flavor of this localization of function. But whether it's through study of patients with brain lesions, whether it's through electrical stimulation or electrical recording, or whether it's through these fancy new brain imaging techniques, we're always looking in psychology to correlate structure with function. What's going on in the brain when people are engaged in some particular form of mental activity? Here is a classic case in which brain lesions were used to infer the localization of certain psychological functions. This is the famous case of the patient H.M., who suffered, a young man, about 18 years of age, who suffered from very severe, intractable epileptic seizures. These were seizures they were very frequent, they were very severe, they weren't controllable by any of the medications that were available at the time. Through use of a recording technique called the electroencephalogram, or the EEG, HM's physicians localized these epileptic seizures as arising from the temporal lobe in his brain, and as an act of desperation, they decided that they would just take out some of the temporal lobe in an attempt to alleviate his seizures. It's a radical piece of neurosurgery. Everybody said, well, lots of people said, don't do this. Nobody knew what these structures did at the time. They found out when HM woke up from the surgery, because what the surgeons did was to remove the interior portion of the temporal lobe. Remember, frontal, parietal, occipital, temporal lobe. The interior portion of the temporal lobe, including a subcortical structure known as the hippocampus. And when HM came out of the surgery, to some extent the operation was a success. He no longer suffered these same kinds of epileptic seizures. They apparently had excised the area of the hippocampus, of the temporal lobe, that where these seizures were being generated. But HM no longer had any memory. That is to say, he was not able to remember anything that happened to him since the time of the surgery. He was able to remember his pre-surgical life fairly well. There's some controversy about that, but mostly he was able to remember his pre-surgical life. But his post-surgical life, what the doctors call his post-morbid experience, was completely lost to conscious recollection. This surgery was in 1953. HM is still alive. He's living in an extended care facility in New England, and he has no conscious memory of anything that has happened to him, of anything he's experienced, anything he's done since the day of the operation, leading people to suspect that this, we now know that this structure, the hippocampus, which is lodged inside the interior portion of the temporal lobe, is very critical for laying down memories in such a way that you can get conscious access to them later. The hippocampus isn't the storehouse of memory, but the hippocampus seems to perform an important function in encoding and indexing memories so that we can get at them later. We know that from studies that have been done on patients and animals since the HM case. But here's an instance where HM, a lesion in HM's brain, led us to new insights into the relationship between structure and function. Question? Yeah, they took it all out. This whole shaded area here, the interior portion of the temporal lobe, and including the hippocampus, the amygdala, some other structures here, they just went in and scooped it out. Okay? You got to remember that this was an act of desperation on the part of everybody. HM didn't want to live with these seizures anymore, okay? They're terrible seizures, just really, really, really terrible. And there was no other way to control them. It was an act of desperation. No, we don't do this surgery anymore, right? But at the time, nobody knew what the function of the hippocampus was. Nobody had any idea that it had any particular function. If you think about it for a second, you say, well, it's there, it's got to do something, right? But nobody knew what it was, okay? People generally thought that important mental functions were performed by the cerebral cortex so that you could take out some of these subcortical areas and not do any harm. We know better now, but they didn't know that then. Yeah, he lost quite a bit of his temporal lobe, not the whole thing, for reasons I'll talk about in a second. Was that the answer you were looking for? Oh, okay. Well, there's a device called the electroencephalogram that you can attach electrodes to the skull and record the electrical activity of the cortex underneath that electrode. There's just enough electrical activity that you can kind of pick it up going through the skull and the scalp. So they put a whole bonnet of these electrodes on HM and waited for him to have a seizure. Okay, well, what are you going to do? That's how you do diagnosis, right? They waited for him to have a seizure, and when they noticed him having a seizure, they noticed that the seizure activity, which is very clearly visible on the EEG, you recognize it immediately, began in the temporal lobes, okay? So that led them to think, correctly as it turned out, that there was something wrong with that portion of the temporal lobe that was causing this seizure to be generated and then spread to other parts of the cortex. So they thought, all right, if the seizure is starting out in the temporal lobe, let's chop out some temporal lobe, okay? And we'll get rid of this. They were right, okay? And and we wouldn't do the surgery again, but they didn't know any better. You have no idea how primitive neurosurgery was just 50 or 60 years ago. But as I say, there's one of these great and desperate cures for a condition that was otherwise not susceptible to any kind of treatment. Yes? Would the amount of damage be correlated with the amount of tissue they took out? That's a general principle, but it turns out it's not the amount of tissue you take out, it's exactly what part of tissue you take out. Here's the hippocampus right in front, here's the amygdala, there are some other structures here. It's the hippocampus. If they had taken out his temporal lobe and left his hippocampus intact, he would have been free of his seizures and he'd have his memory. But that's very, very, very difficult to do. And given the surgical techniques of the time, you can't really separate the hippocampus from the rest of the medial portion of the temporal lobe, not without very, 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 very careful surgery of a kind that they just weren't prepared to do. Again, if they had just left the hippocampus alone, everything would have been hunky-dory, okay? But they had to take the hippocampus at the same time. They didn't know it was going to cause a problem, so that's what they did. Okay, but there are cases where people have lost. Remember that there are two sides to the brain. You got two hippocampi, one left, one right. If you take out one hippocampus and not the other, you still have some memory function. It's bilateral resection of the hippocampus that obliterates conscious recollection and creates this syndrome. Yes, one more question in the back. We're going to get to that in a minute, okay? And that's why they didn't take the whole temporal lobe out, because it actually does some interesting things. Anything else on HM for a second? Yes? Did they do this procedure more than once? Not too many more times than once. Well, what they did mostly was monkeys, okay? No, they wouldn't do this again. It takes a while for word to get out, okay? But HM was such a unique case, okay? I don't think there's another case of deliberate, unknowing surgical excision of the hippocampus. Word got out pretty fast that you shouldn't do this. And of course, half the neurosurgeons in the country said to the surgeon, "Oh, you're crazy, don't do this." But he said, "Look, we don't know." You know? He's got these seizures. He doesn't want these seizures. HM gave informed consent given the fact of what we knew about the brain at the time. Okay? It's important not to kind of impose our current knowledge of the brain on these guys. They just didn't know, okay? And nobody could have told them that the hippocampus had these functions. One more question, then I got to go on. Yeah, I'm going to talk, no, he did not lose other functions in the temporal lobe for reasons I'll talk about in a minute, okay? If I don't answer that, ask me again. I talked a little bit about the amygdala before. Here is another patient, SM, a woman who suffered calcification of the amygdala, which sits right in front of here's the temporal lobe, here's the hippocampus buried in it, and right on the front of the hippocampus, kind of like a fender or something, is a structure known as the amygdala. This woman had a very, very rare syndrome known as bilateral calcification of amygdala. And basically what happened is that the amygdala, it calcified. It turned to essentially bone tissue, dried up, and turned to something very much like bone. And it affected only the amygdala. This calcification did not extend into the hippocampus, and it didn't extend anywhere else into the temporal lobe. This is a very rare neurological syndrome, but you do see cases of it from time to time. And what happened in this case is that patient SM basically had no cognitive impairments. She didn't suffer amnesia the way HM did because her hippocampus isn't damaged, but she had problems in her emotional life. She had a lot of difficulty experiencing negative emotions, especially the emotion of fear. And she had a lot of difficulty recognizing other people's emotional states from their facial expressions. We recognize this immediately as a person who is in a state of fear, but SM neither experienced fear nor did she recognize fear in others. In much the same way that we infer the memory function of the hippocampus from what happened in cases like HM, we infer the emotional structure of the amygdala from what happened in cases like SM. It turns out that the amygdala plays a critical role in regulating our emotional lives, both the experience and the expression of emotion, particularly the negative emotions and particularly the emotion of fear. So here we have two very good examples of how we can use lesion evidence to make inferences about the functions of various kinds of structures. Questions about SM? Yes? No, she doesn't experience negative emotions. Sorry. She doesn't experience much emotion at all. But she especially is impaired in terms of negative emotions and especially in terms of fear. The amygdala is sometimes known as a fear center in the brain. Yes? People with autism have different sets of problems. We're going to talk about that a little bit later on. But this is not autism, okay? And autistic individuals don't have particular problems with the amygdala. They have problems in experiencing and expressing emotion sometimes, but that's part of a whole package of other kinds of problems that autistic people have. To my knowledge anyway, we've not been able yet to locate any discrete brain damage that's reliably associated with autism. There is something wrong in the brains of autistic people, presumably, but it's nothing like a discrete lesion in the hippocampus or the amygdala or any place else. One more question, then I got to go on. Well, SM, since her memory wasn't impaired, can she remember fearful experiences? I got to answer that two ways. The first is that she can remember fearful experiences that occurred to her before her amygdala got calcified. She can say, oh yeah, I remember going up in this tram once on a ski slope and I was looking down and I was really afraid I was going to fall. She can remember that. She cannot reproduce that experience the way you can if you're telling a story about a time when you were afraid. You actually begin to feel afraid again. She doesn't have that experience. Okay. Second point, something that'll be more understandable later on, she can't acquire new fear responses. Okay? So she can't learn to fear something that she didn't fear before the damage occurred. Because she doesn't experience fear now, so she can't learn about fear, right? What she can do is to recognize objects that, how to put it, she can recognize objects that she ought to be afraid of. Okay? So you can show her a picture of a rattlesnake if she had never seen a rattlesnake and say, look, when you're mucking around in the desert, stay away from this. Okay? And she can learn that. She says, oh, okay, I will stay away from this. But she doesn't have the fear experience that would be the basis for most of our learning about this. She can learn about things to be avoided, things to be feared in the abstract, but these are not accompanied by any experience of fear. The amygdala seems to be important in generating that experience. When the amygdala is gone, the experience is gone as well. Okay? So there you go. Two examples of how lesion evidence can be used to infer the structures of various pieces of the brain. Quick question. What happens to the physical effects of fear, like sweating and pupil dilation? They're generated by the amygdala. Her amygdala is gone. She doesn't have those physiological effects. Okay? They're just not part of her behavioral repertoire any longer, precisely because it's the amygdala that generates those physiological responses. Okay, turning attention to the cortex itself, get back to a couple of other questions here, especially about the temporal lobes. We can get other evidence of structure-function relations by looking at a, unfortunately, large number of patients who have suffered from a neurological syndrome known as stroke. Strokes are basically heart attacks that occur in the brain, not in the heart. There is a vascular insufficiency, an insufficient blood supply to the brain. Blood isn't carrying the oxygen the brain needs to stay alive, and so certain structures in the brain die. This is a rough diagram of the vascularization of the brain, and you can see that there are these big blood vessels that run up along the central fissure, carrying lots of blood to various parts of the brain. When people suffer from a stroke, a vascular insufficiency or an insufficient blood supply to the brain, there are three common symptoms that we observe. The first of these is paralysis, a loss of voluntary motor function. Somebody down here in the last lecture asked about hemiplegia. Hemiplegia is something that happens with strokes, loss of voluntary motor function, okay? Or anesthesia, especially tactile anesthesia, a loss of tactile sensation in various parts of the body, another prominent symptom of strokes. And then finally, a neurological set of symptoms known as aphasia, basically having to do with a loss of speech and language function. And what happens now is that, especially by virtue of examining stroke patients and where the damage is occurring by virtue of an insufficient blood supply to the brain, we can infer additional aspects of structure-function relations. So, for example, we now know that there is a portion of the frontal lobe just adjacent to the central fissure. Here's the central fissure. Here's the lateral fissure, frontal lobe, parietal lobe. There's a portion of the frontal lobe that is known as the primary motor area, and this appears to be an area that generates voluntary motor behaviors of various kinds. When some or all of this portion of the brain is damaged, okay, because no blood's getting to it, then you lose some voluntary motor function. By the same token, there is a part of the parietal lobe just adjacent to the central the fissure that is called the primary somatosensory area, and this seems to be an area that processes tactile sensation from various parts of the body, the sense of touch. So damage in this area will result in the loss of tactile sensation in various parts of the body. Again, we know this primarily from stroke patients. There's other evidence as well that suggests that here in the frontal lobe, we have a part of the cortex that's devoted to generating motor activities, voluntary motor activities, and we have a part of the parietal lobe that is devoted to receiving tactile sensations from various parts of the body. In fact, there's quite a bit of interesting organization to both the frontal motor area and the parietal somatosensory area, and this can be illustrated by what's known as actually two structures, the sensory and the motor homunculus. Homunculus is just a little Latin word, means little man, okay? This is the little man in the head, and Wilder Penfield, the great Canadian neurosurgeon, used this as a way of illustrating what parts of the motor and somatosensory areas are associated with what functions. So here, imagine now that we've sliced the brain down along the central fissure, and we're now looking at it from inside. Here is the cerebral commissure that divides the brain into two hemispheres. So inside the cerebral commissure, you've got it here, and then running right down the central fissure here into an area right tucked up next to the lateral fissure, which is what this is. Okay, in the frontal lobe, okay, what we have are various areas of the frontal lobe that are dedicated to various parts of the body. So here, in the frontal lobe, in the area tucked into the central fissure, is a part of the frontal lobe that controls the motions of the toes, the feet, the lower leg, the upper leg, the torso. Now we're coming out from the interior portion of the brain, down the side of the frontal lobe, right next to the central fissure, part of the motor cortex that controls the upper arm, the lower arm, the hand, the fingers, the face, the brow of the head, the face, the lips, the jaw, and here's a portion tucked up under here where the lateral fissure is that controls swallowing behavior. The motor activity of each different part of the body seems to be associated with control by a different portion of the frontal lobe. Similarly, with the somatosensory areas of the parietal lobe, there's a different part of the somatosensory cortex associated with each individual part of the body. So here, tucked into the central commissure, is a part of the area that receives tactile sensations from the genitalia. Then the toes, the feet, the lower arm, the lower leg, the upper arm, the torso are now running down next to the central fissure in the parietal lobe, the upper arm, lower arm, hands, fingers, tactile sensations in the face, the eyes, the nose, the face, the lips, here in the teeth, the tongue, the pharynx, and so on, tucked here under the lateral fissure. Notice two principles that we talked about when it came to the spinal, to the spinal nerves. First is a principle of regular association. The part of the frontal lobe that controls the movement of the fingers is right next to the part of the frontal lobe that controls the movement of the hand. The part of the frontal lobe that controls the movement of the lower leg is right next to the part of the frontal lobe that controls the movement of the upper leg. Similarly, for the somatosensory cortex on the parietal side. There's a regular association between the part of the body that's being moved or being sensed and the part of the brain that's doing the movement or doing the sensing. The other feature you should notice here is, again, a feature of proportional representation. In the frontal lobe, the amount of frontal motor cortex devoted to the various parts of the body is proportional to the need of that body part for fine motor control. You need very fine motor control in your fingers. Look at how much cortex is devoted to your hand and your fingers compared to the trunk of your body, which is by any standard much larger. Okay? Similarly, in the somatosensory area of the parietal lobe, proportionally more parietal cortex is devoted to those body parts where you need fine tactile sensitivity. Again, in the hands, in the face, in the feet. Where you want a lot of tactile sensitivity, you've got a lot of tissue devoted to that body part. So as with the spinal nerves, we see again these two principles of organization come into play. The regular association between some part of the brain and the part of the body that it serves, and a proportional association between some part of the brain and the body that it serves. Two more things. We also have here in the temporal lobe what's known as the primary auditory area, the lateral portion of the temporal lobe. We're looking outside, from the outside in now. Here is a part of temporal cortex that's devoted to auditory processing, to hearing, and so on. And here, the very back of the occipital cortex is a part of the brain that is devoted to visual processing. When somebody experiences cortical deafness, there are lots of different kinds of deafness, but cortical deafness typically involves damage to this portion of the temporal lobe, which is the primary auditory area. Cortical blindness, there are lots of different ways to be blind, but cortical blindness typically involves damage to this primary visual area in the occipital lobe. So again, we have, do I have this? Okay. There are lots of different visual areas as well. Don't worry about that. Okay. But basically, what you've got here are illustrations of the principle of localization of function. Motor function in the motor strip of the frontal lobe. Somatosensory function in the somatosensory area of the parietal lobe. Auditory processing in a piece of the temporal lobe. Visual processing in a piece of the occipital lobe. Something about memory in the hippocampus, something about emotion in the amygdala. That's the general story behind localization of function. We'll complete it on Monday. Thank you very much.